Good afternoon. Today is Sunday, June 14th, 2020. My name is Ricardo Sandoval, and this is the results video for Lab 8, Biochemical Testing, Carbohydrate Fermentation for Biology 225, Microbiology at Los Angeles Pacific University for the Summer 1 term. This is just a quick little sign, just summarizing what I just said. So, not focusing very well, but to recall what I did. So I had 6 tubes of a water-soluble dye called phenol red that was also mixed with a bit of nutrient broth, and then I had 3 sugars that act as carbohydrates. So I had glucose, fructose, and mannitol. Again, glucose is what's used in energy for ATP production in cells. ATP is adenosine triphosphate. It's also what we use to create energy. Fructose is what sugar produced by fruit. It's what makes fruit sweet. And then, of course, there was mannitol, which is a sugar alcohol produced by most fruits and vegetables. So I had glucose, fructose, and mannitol, which are all simple carbohydrates. And then with 6 tubes, so 2 of the tubes got fructose, 2 of the tubes got glucose, 2 got fructose, and then 2 got mannitol. And then each of the tubes got something called a Durham tube, little test tube looking things, very small, that are used, that are in each tube face down with the opening down, to see if any gas is developed during the fermentation process. And then each of the pair of tubes got either 4 drops of the S. epidermidis or the S. cerevisiae. And then the tube sat for a total of 13 hours to ferment, to determine if the solution itself was a fermenter, a fermenter with acid and gas, or a non-fermenter. So again, the big thing here is fermentation. And what fermentation is defined as is it's the conversion of something called pyruvic acid. And pyruvic acid is a byproduct of a process called glycolysis. And then once fermentation takes place, we either get an acid by itself or an acid with gas, this is why we have a fermenter by itself and a fermenter with acid and gas. And then common end products of the fermentation process, they include lactic acid, formic acid, acetic acid, butyric acid, ethyl alcohol, carbon dioxide, and hydrogen. And again, just to touch on something I mentioned in the experiment video, let's think of champagne again. So I added sugars to my tubes here to start fermentation, or to create fermentation. And in the same respect, champagne starts as plain white wine using champagne grapes from the Champagne region of France. But to cause the fermentation that causes the bubbles that makes champagne bubble and foam when you pour it, sugar is added to cause fermentation, which is the same principle here. That being said, I'm going to start showing you my test tubes here. So I labeled my test tubes number 1, number 2, and number 3. So number 1 is for glucose, number 2 is for fructose, and number 3 is for mannitol. And then all of these tubes, before I added anything, started out a reddish color, which was the phenol red. So here we have the number 1 S. cerevisiae glucose. And then as you can see up here, we have some foaminess, and then there's the Durham tube, and then you can see some bubbles here. So since we have this yellow color here, and the foaminess, I can conclude that this is now acidic, and with the foaminess up here and the bubbles in the Durham tube, we have a fermenter with gas. And then I know this is acidic because if we look at the pH scale, which is right there, that yellow color is on the lower end. on the acidic end. Now, one thing not to be confused about here is that red does appear on the acidic end as well, but phenol red is red. And in its unaltered nature, before I added sugar, before I added any of the bacteria, its pH is around 7, so it's neutral. So the red in and of itself does not indicate any sort of acidity. So again, the S-cerevisiae glucose is a fermenter with gas and with acid. Alright, so now we're going to look at the S. epidermidis with glucose, which is here. So there we go. S. epidermidis glucose. And as you can see, this pretty much looks very similar to the identical phenol red that I poured in initially. And there's my Durham tube, which is pretty much, it's flat. It's still untouched. There is a bit of gas there, but this tube has sat for excess of 13 hours. But this S. epidermidis, because the color itself hasn't changed, and there are no real bubbles in here, this is considered a non-fermenter. There's no change in pH, so there's no fermentation, no gas, nothing happened here. Alright, so now we're going to move on to fructose with the S. cerevisiae. So again, here is number 2, S. cerevisiae fructose. And again, we have the yellow color. So again, looking at the pH scale, we have yellow. But the Durham tube itself is kind of at the bottom. There does seem to be some gas here, but not much going on. And the Durham tube itself isn't floating. I don't see any clear gas bubble in there. And again, but there is a little light layer of foam, if you can see up here. So I can conclude here again, this fermented, it is acidic, and we can see, there is a small amount of gas, but the Durham tube itself was placed in there to see if it would float with air bubbles, and it didn't happen. So this is a fermenter, perhaps. It is definitely acidic, but perhaps without any sort of gas. Alright, so the next up is the S. epidermidis fructose. And there we go, S. epidermidis fructose. There's my Durham tube. So you can see some little bubbles in there. But, I mean, the tube itself is at the bottom. The color of the phenol red has not changed from its original state. So, just like the previous S. epidermidis, this is a non-fermenter. Okay, so no fermentation took place. Right? And then lastly, we're going to look at the S. cerevisiae with mannitol. And again, we have the yellow color, so this definitely did ferment. It is acidic. The Durham tube is there. And, not many clear bubbles in here, but you can definitely see a nice layer there. And there's foam up on top. So, the S. cerevisiae with mannitol did ferment. There is some minor gas production. And then lastly, the S. epidermidis with mannitol remained the same, pretty much the same color of the phenol red before I added anything to it. The Durham tube is there, and there are some bubbles there, but the Durham tube itself is still clearly at the bottom of the tube. No floating, no foam, no turbidity. Nothing really happened here. So S. epidermidis mannitol is also a non-fermenter. So, since I used only 2 microbes, S. cerevisiae and S. epidermidis, and all 3 of the S. epidermidis, of the S. cerevisiae tubes, which are here, fructose, mannitol. Oopsie, I want to turn this one, or this one, I should say. Whoa. Didn't break, so that's good. So, fructose, glucose, and mannitol are all yellow. It's safe to conclude that S. cerevisiae definitely is a fermenter, and these two organisms for sure. And these two organisms, which are glucose and fructose, there's definitely some foam here. And in the mannitol one, there's also a little tiny bit. So, but S. cerevisiae itself is a fermenter. And then in looking at all three tubes of the S. epidermidis, fructose, glucose, and mannitol, no fermentation took place, no change of pH took place. So S. epidermidis in and of itself is not, it doesn't ferment. It's not a fermenter even with the addition of simple sugars or simple carbohydrates. I did not encounter any problems with this laboratory. It was relatively straightforward, and it was quite unique to see how the fermentation process does take place. This concludes the results video for Lab 8, Biochemical Testing Carbohydrate Fermentation. Again, my name is Ricardo Sandoval, and this video was for week 5 at Los Angeles Pacific University, Biology 225, Microbiology for the summer 1 term. Thank you.